Okay, so today's video is going to focus on genetics. We're going to do a lot with vocabulary, and then we're going to look at how to do a simple monohybrid genetics prediction problem. Okay, so we're going to start with a little bit of vocabulary review here. We've talked about genes before. Remember, a gene is just a section of DNA that codes for one particular protein. And remember, we get to that protein by going through the central dogma theory, right? We go through DNA to the messenger RNA, which is our transcription, and then the messenger RNA to the protein, which is the translation. Alleles are just going to be different variations of the same gene, meaning remember that the gene is coding for one protein. So alleles are different variations that are coding for that same protein. If you look at the picture in the example, it looks like all those proteins are the same, just different colors. So you could say that those alleles, the different alleles is what gave you a green protein or a purple or a blue. Okay. Alleles can be represented as dominant or recessive. Dominant alleles are usually represented with a capital letter, recessive with a lowercase letter. So big A versus little a here. Okay. Dominant genes are always expressed. So if I had an allele that was dominant, it would always show up. It would be expressed as in we'd be able to see it. Recessive alleles are not always expressed. They are sometimes essentially hidden by the dominant allele, by the dominant gene. You can only see the recessive alleles if both copies, both alleles have to be recessive for it to be expressed. Let's revisit what we mean by both alleles. If you remember, alleles or genes, our chromosomes are in pairs, right? And so we got one from mom, one from dad, right? We've got one from mom, one from dad. On that chromosome, there may be one little gene here. Let's say that gene is for eye color. Okay? And maybe dad has one version of that gene and mom has a different version of that gene. Let's say that mom has the allele for brown eyes, which is dominant, so a capital B. Dad has the allele for blue eyes, which is recessive, so we write it as a lowercase b. So therefore, your two alleles that you have for eye color is one big, one little, one dominant, one recessive. Your eye color then is going to look brown because the dominant one is going to overshadow the recessive one. And now let's look at the vocabulary that's going to go along with that. Okay, so when we have the pair of genes, that is called our genotype. So let's say I have big A, little a, or big A, or little a. That's going to be referred to as the genotype. The genotype will always have two alleles because it's representing what you got from mom, what you got from dad. Remember our karyotype up here that shows our pairs of chromosomes, our homologous chromosomes put together. So let's say we're looking at this gene, and maybe here it's big A. That's my genotype. My genotype can be described as homozygous or heterozygous. Remember, homo is same, hetero is other or different. So if I am homozygous dominant, my genotype is big A. That's what I have right here. If I'm heterozygous, other or different, That means I have to be big A, little a. That, again, is my genotype, the actual alleles. So then for homozygous recessive, both letters have to be the same, and they're the recessive, they're the lowercase. The phenotype is going to be the physical. What is actually expressed? What does it actually look like? So at the top, again, for this homozygous dominant genotype, this big A genotype, this bird looks red. It's got one allele for red, one allele for red. It looks red. In the heterozygous, it's got one allele for red, but it has one allele for blue. However, the phenotype of the bird is red because red is dominant, and so red is masking that blue color. We'll talk later about what it looks like if it doesn't fully mask it. The phenotype for this final bird here is going to be blue because it has two recessives. There's no dominant. So the only way to get a recessive phenotype is to have the genotype be homozygous recessive. That's going to be really important later in certain descriptions of things and such because it allows you to narrow it down. So again, let's reaffirm that there. The only way I can get a recessive looking phenotype is if the genotype is homozygous recessive. So let's look at how we make these different predictions for what the offspring will have. So we're going to do an example of a monohybrid. So we're referring mono is one. So we're only looking at one trait in these examples. And we're going to use a Punnett square to predict what the offspring will look like for these two individuals. So the initial cross is between these two parents, which is a big G, which is a true breeding. Sometimes that is called a purebred green pod, and a true breeding or a purebred yellow pod. Remember, if I had just given you the description of green crossed with yellow, green could have been big G, or big G, little g. Both of those would look green. I know it's big G though, because it says it's true breeding. For it to look yellow, which is recessive, the only genotype it could have is little g. That's how that starts to become helpful to us to keep that in mind. Okay, so when we set these monohybrid crosses up, we take big G, or we take the one parent, and we take the gametes that parent could produce. So they could possibly have a gamete that has a big G, and they could possibly have gametes with a big G. That's all they can pass on. So I take those gametes and I put them on one side. I don't mix them up with the other one. I put them on one side. Then I do the same for the second parent. They can produce a little g gamete or a little g gamete. So I put their gametes over here on the side. Now I just bring them together, like a regular grid, right? Big G, little g, big G, little g, big G, little g, big G, little g. Inside the circle here, these are all my possibilities for the offspring that these two plants could produce. Those are my possible genotypes. So if I described these offspring, I would say that I'm going to have a big G, little g. I'm going to have a hybrid. That's what a big G, little g, or a heterozygous. That is a hybrid. So the offspring are all going to be hybrid, okay? And because they're going to be hybrid, they're going to look green. These two plants should not be able to produce any yellow pods. So when we do these Punnett squares, there are 3 different ways that we can write these genetic predictions that we get of the offspring. So the first way would be to write it as a fraction. So in this example over here on the left, right, I had a cross of big G crossed with big G, little g. So I could write this as a fraction. For instance, let's see, how maybe it asked about heterozygous genotypes or hybrids. What fraction would be hybrid? So 2 out of 4 or 1/2. So I can write it as a fraction. I can write answers to questions as a percentage. So in this one here, I had 2 hybrids were crossed, and here's my possible offspring. Maybe it wanted to know the percent that would be yellow. Well, yellow was the recessive phenotype, so only this one right here could look yellow, so 25%. So it could ask me about phenotype, it could ask me about genotype. I can also look at it from a ratio point of view. So in this final example here, where we had this cross here, maybe I wanted the ratio of green to yellow. So the ratio of pods that look green to the ones that look yellow. Well, this one and this one will look green with the big G's. So the other two will look yellow. That reduces down to a 1-to-1 ratio. We're going to look closer at ratios in the next slide. But fractions, percentages, decimal places, ratios, these are all options. So you need to slow down and read the question and make sure you know which one it's asking you for. So there are 2 types of ratios that you will need to be comfortable with for monohybrid examples, genotypic ratios and phenotypic ratios. So let's look at this particular example here. I'm crossing 2 hybrid organisms. So I take one parent, the possible gametes that they could produce are big G and a little g. Remember, gametes are egg and sperm cells, so they have the haploid number of chromosomes, so they have been reduced in number. So the other one could produce a big G or a little g. So when I solve my square, these are my possible, not guaranteed, my possible offspring combinations. So when I do genotypic ratio, I'm looking at what are the possible genotype ratios compared to one another. So my possible genotypes, right, are big A, homozygous dominant, heterozygous, homozygous recessive. So homozygous dominant to heterozygous to homozygous recessive. That order, every time we do the ratio, that order stays the same. If you have a 0, go ahead and put the 0 in to hold your place. So all I have to do this ratio is just count them. So the homozygous dominance, I have 1 that is homozygous dominant, so 1. I have 2 that are heterozygous genotype, 1 big G, 1 little g. And then I have 1 again that is homozygous recessive. That's my genotypic ratio, 1 to 1. That's all I have to do to calculate it. I just need to keep that order every time. Homozygous dominant, heterozygous, homozygous recessive. For phenotypic ratio, remember, phenotype is the physical. It's what's expressed. It's what they look like. So in this case, I only have 2 options. It can look like the dominant trait, which in this case was green, or it can look recessive, which in this case was yellow. So I'm going to do that based off of the phenotypes. And based off of the phenotypes, I have 1, 2, 3 that will look dominant. All 3 of those will look green. Remember, because the big G will mask the little g. And one of those, this one at the very end, that's the only one that's going to look yellow because it has 2 recessives. So my phenotypic ratio here is 3 to 1. For monohybrids, genotypic ratio should have 3 numbers because there's 3 possible genotypes. Phenotypic ratio should have 2 numbers. We'll look, there's going to be some exceptions to that in the phenotypic world when we move into some non-Mendelian patterns, but we'll worry about that when we get there. Okay, so as always, make sure that you have made notes of your questions so that we can answer them in class, because the expectation would be, right, that you can do these examples and you can solve monohybrids so that we can, do a bunch of examples and do some activities with that in class.